Hi, my name is Nilsa La Cunza, and I'm a fifth-year graduate student at the University of California, San Francisco. I work in the ophthalmology department, and my project focuses on understanding how a cholesterol transporter is associated with a blinding disease called age-related macular degeneration. Now, AMD is a very complex disease. Over 50 independent genes are associated with the disease, and these control many different pathways, including cholesterol dysregulation. I chose to study this cholesterol transporter, apolipoprotein E, or ApoE, because it's a very interesting protein. All the species studied so far, except for humans, express only one type of this protein. The protein that most species express is similar to ApoE3, and about 78% of us express ApoE3. But around 200,000 years ago, our ancestors branched off and started making two other kinds of this protein, called ApoE2 and ApoE4. Now, what's really interesting is these three isoforms differ in one amino acid from ApoE3, and this one amino acid difference can significantly affect how this protein functions. If you look at the affinity for the LDL receptor, ApoE3 and ApoE4 have high affinities, and ApoE2 has low affinity. And that dictates how well it transports cholesterol in and out of the cell. Now, what's interesting for us is that ApoE4 is a confirmed risk factor for Alzheimer's disease, while ApoE2 is protective. And this risk is flipped when it comes to age-related macular degeneration, or AMD, where ApoE4 is protective and ApoE2 is the risk factor. So we noticed in our experiments that ApoE vesicles and retinal pigment epithelial cells, or RPE cells, which we study, would aggregate into clumps, which we could not explain until we thought of liquid-liquid phase separation, a novel phenomenon that has been implicated in many neurodegenerative diseases such as ALS and Alzheimer's. Liquid-liquid phase separation is a mechanism by which proteins or liquids can be condensed into aggregates. If proteins with intrinsically disordered regions are stressed or undergo post-translational modifications, it can influence liquid-liquid phase separation, and thus persistent stress can regulate the formation of these aggregates called biomolecular condensates. So we know from other studies in Alzheimer's that ApoE has intrinsically disordered regions. So perhaps these ApoE clumps that we're seeing are actually drusen aggregates, since drusen are composed of apolipoprotein E, and they are a hallmark of the disease. So we asked, could abnormal ApoE phase transitions contribute to drusen nucleation in the RPE? I did see a difference in aggregate volume. So these are primary RPE cells that have been transfected with the human ApoE2, 3, and 4 isoforms, as you can see on the top layer in red. And the bottom are 3D reconstructions of these ApoE vesicles, where the warmer colors represent larger aggregates and the cooler colors represent smaller aggregates. So we can observe that ApoE2 expressing RPE cells have larger aggregates than ApoE3 and ApoE4. Now, another characteristic of biomolecular condensates is responding to changes in the intracellular environment. So I wondered whether declining the metabolic activity in ApoE2 RPE cells due to the loss of mitochondrial function could increase chances for aberrant phase transitions. I transfected primary RPE cells with the three ApoE isoforms and exposed them to two stressors that are implicated in AMD: excess cholesterol and complement activation. I analyzed the mitochondria by creating a surface reconstruction of the mitochondrial network to define whether it was interconnected and healthy or fragmented and unhealthy. In this reconstruction, cooler colors represent unhealthy mitochondria and warmer colors represent healthy mitochondria, as you can see below. This resulted in mitochondrial fragmentation of the ApoE2 expressing RPE cells. Now, our study elucidates the relationship between the ApoE genotype and pathogenic triggers in the RPE.